Morning, students.
Good morning, sir.
Yes, sir.
So let us continue talking about biotechnology tools. First of all, we were discussing. So last class we understood about, in brief, what is basically biotechnology and what type of activities are carried out. In brief, it is basically nothing but a process wherein we can do manipulation of the genetic material in an individual. So we can change the genetic makeup of an individual to a certain extent. But most important requirement for this is you cannot simply do anything you want. Whatever manipulation you are looking forward, that must be beneficial for human beings in some or the other way. And that is why these manipulations are carried out, for which various tools are required, and different tools. Among them, the first one that we discussed was the tool for cutting of the DNA, so that we can take out a gene of interest, a specific gene of interest, or in certain places you will also learn later on in the process that in certain steps you sometimes need to open up a particular DNA molecule. So there also this enzyme, restriction endonuclease enzyme, is used as a molecular scissor, something that can cut the molecule. Now, today, the next few tools that we are going to talk about, after understanding about this restriction endonuclease enzyme, is second thing, Agarose gel electrophoresis. Okay. Now here, first of all, this is a technique which is mainly carried out to separate the DNA molecules. Okay. Let us say you have taken a DNA molecule initially, and that DNA molecule, suppose you have cut down with the enzyme into multiple pieces. Okay, let's say you have cut it down into 1, 2, 3, 4, 5 pieces, for example. And now your requirement is, out of all these various pieces of DNA, you are looking forward only this particular piece. Now, if we look at it very practically, how it is done, you will take the DNA, you will isolate the DNA, and you will have the DNA in a buffer. In a test tube, you have the buffer. So in the buffer, you have here the DNA molecule. Now this DNA molecule, when you have put the restriction endonuclease enzyme in it, then it has broken down into multiple pieces here. Now this buffer, which is there in this particular tube, it contains lot of DNA fragments. Now our idea is, out of all these DNA fragments, you have to isolate the specific, what we call as gene of interest. Okay. So now how will you separate out of all the DNA fragments? How will you separate this gene of interest? So that process is this process which is called as gel electrophoresis. Okay. First of all, let us understand what is the principle behind this particular process. Now here, the main word is electrophoresis. As you can see, we have broken down the word. Electro means basically a electric current, flow of electricity. So there is an electric field which is created in a gel slab. Okay. And phoresis basically is kind of movement to carry across. Okay. Or there is a movement of molecules through this gel. Now, what is this gel? Gel is a colloidal suspension, okay, which is made up of many small particles in a medium, and it forms a semi-solid substance like gelatin, right? As a result, what happens? You will get a slab of gel, something like this. You will get a gel slab like this. And in this gel slab, there will be many small, minute pores that will be formed here, right? Through these minute pores of the gel, we allow the DNA molecules to move, right? Understand? I will show you a diagram. Diagrammatically, it will become more clear. This one you see. This is the actual, what we say, kind of setup. Okay, but this is a schematic diagram which explains what we do is there is a chamber, right? So this is a chamber here where we can take certain buffer. Here, buffer is taken. Buffer is taken in such a way that it will allow electricity to pass through it. So you can see there are two electrodes. This is a negative electrode and this is a positive electrode on two sides. And then in this buffer, there is a submerged gel slab. This is the gel slab here. In the gel slab, we have made a facility, a well is made. We call it as a well, where the sample is loaded. Now, what do you mean by sample? A sample is basically nothing but the DNA fragment mixture. This is a mixture of DNA fragment. Okay. Now, the DNA molecules, when it will be here, and we switch on the electric current, then there is an electric field which is generated. Okay. Electric field is generated. Now, since the DNA is negatively charged molecule, so we have put it towards the negative electrode. And because of the electric field, it will be attracted towards the positive electrode. So these DNA molecules, they will start moving through this gel, through inside this gel, towards the electric, towards the positive electrode. Okay. Now, this is what is called as electrophoresis. Under an electric field, we have made the DNA molecules to move forward. Now the point is, how does it help? Because this gel slab has very minute pores in it, and the DNA molecule is passing through these pores. So based upon the size of the DNA or the molecular weight of DNA, what will happen is the smallest fragments will move the largest distance, and the largest fragment will move the least distance. Okay. So, understand this diagram. Take any one sample. Let's say sample A. It was loaded here, having the various DNA fragments. So this side was a negative electrode. This side you had a positive electrode. So this is your gel slab. Now when the DNA molecules will move, these bands which you can see, these are called as the DNA bands. Okay. Means as of now, just understand that these are the positions where certain DNA fragments get settled. Okay. What are bands and how they are formed, I'll tell you later. First understand, DNA fragments will get settled at different positions. So here you can say, therefore, the DNA which you will get in this band will be the largest fragment DNA. And the DNA settled in this band will be the smallest fragment DNA. Just for example, I can say something like, this is a 2 kb DNA, 2000 base pair DNA. Okay. And this one is let us say 4 kb DNA. This is 8 kb DNA. This is 12 kb DNA. This is 13 kb DNA. Just for example, this is suppose 16 kb DNA and this is suppose 20 kb DNA. Kb means 1000 base pair. Okay. So based upon the size of the DNA fragment and therefore their molecular weight, because smaller the size, lesser will be their molecular weight, larger the size, greater will be their molecular weight. So the DNA here got separated. This separation of DNA which took place, the separation of DNA fragments, okay, it is based on what? It is based on the size of the DNA fragments or the molecular weight of the DNA fragments. Clear? Is the principle understood? So this is the facility, this is the actual, you can say, electrophoresis apparatus, right? This is another picture where the process is being carried out. This is a power supply setup. Okay, you can see here they have put one electrode here and another electrode here, right? This is a chamber where if you see this diagram, second diagram, this is a whole chamber where there will be a buffer, and this slightly translucent part which you are seeing, this is a gel. This is a gel slab. Okay, and you can see within the gel slab these regions. Okay, this is where the DNA is being loaded. This is the technique. This is called as a micro pipette. You have seen pipette in your chemistry lab. This is called as a micro pipette. This can hold very small amount, like few micrograms, few micro mLs, like that. Normal pipette you have seen, they will have usually a capacity of minimum of around 1 ml. 1 ml pipette you might have seen. This is something which can hold more substances to up to like 1 microliter, 2 microliter, like that. Very small amount. So we call it as a micro pipette. So with the help of this micro pipette, this DNA is being loaded into these small wells which are created in the gel. Okay. Then comes, when you put the DNA here, you can see a slight blue color here. Okay. So here actually how this happens is, along with the DNA, now DNA does not have any color. Along with the DNA mixture, they add a loading dye. It's a dye. We call it as a loading dye because it helps us to see or visualize whether the DNA is successfully loaded at a designated well. Because otherwise DNA does not have any color. So even if you are loading something into this, because gel is transparent, buffer is simply water with certain electrolytes in it, right? Therefore, when you put the dye in it, then it becomes visible, and you can see, like here, when this person is loading, he can see the blue color is getting settled at the well, which means that the loading is happening successfully. Now, after this, when it will run, we call it as running of the gel, running of the DNA. So this side must be the negative electrode and this side must be the positive electrode. When you connect it to the electricity power input. Okay, then this DNA from the well will start moving in this direction. When this will move, the DNA fragments, like I have shown in that diagram, starts getting separated. But again, how will you visualize that it has got separated? So for visualization, what is done is a compound known as ethidium bromide. This is a highly carcinogenic product. It is a compound which is used, and this one is actually mixed in the gel. When we make the gel, at that time only we mix it. Okay. It does not have any color of its own. The gel will still look transparent only. But what happens is, when the DNA is present in this particular gel, then there is a complex formed with the DNA. We call it as intercalation. This ethidium bromide intercalates into the DNA molecule, right? After that, when you expose UV light, then it will fluoresce. It appears like a fluorescent dye and will give you a typical color like this. This is the color of this is the picture of a real gel. Okay. This is a gel which is observed against a UV light, right? So below from down, UV light is coming. So from above, we can see this, and then it makes it possible to visualize these positions of the DNA where the DNA is settled, and we call these as DNA bands. Okay, these are called as DNA bands. Try to understand. So here, this fluorescence is observed because the DNA has got intercalated with what we call as ethidium bromide. Okay, so this comes in the exam. How is staining done? Okay, so this is a kind of staining method, like you have learned earlier in making slides and all. What is the importance of staining? Because when you staining is basically a process of providing some dye, and when you stain the cells become visible under microscope. So similarly here, you are not staining exactly, you are not putting a dye exactly, but this ethidium bromide will intercalate with the DNA, and under UV light it will give you the fluorescence and does make it possible to visualize the DNA fragment, right? So this is a kind of indirect staining method. So this is not under microscope, do not misunderstand. There is a instrument, we call it as UV trans illuminator. Very simple, a box like structure, and in the box above there will be a glass, and inside the box there will be a UV bulb. One or two UV bulb will be there. Above that there is a glass slab. On that glass you put this gel, and then the gel will appear like this when you switch on the UV light below. Okay. Achha, one thing I did not tell you. We were talking about, this is called as a gel electrophoresis, but important, that gel is made out of a substance known as agarose. That's why we call it as agarose gel electrophoresis. So this is a polysaccharide. Agarose is basically a polysaccharide which is made from a seaweed. Right? So it is usually dissolved in a buffer, and then you heat it. When you heat it, it becomes a completely transparent water-like, but if you cool it further, then it becomes a gelatinous solid, which will have a network of cross-linked molecules. And thus these network of cross-linked molecules will have within them very small, minute pores. And thus this agarose gel electrophoresis is the technology which is used for separation of DNA fragments. Now the same technology is used for separation of protein also, protein molecules. But in that case, the gel is not made up of agarose, please understand. The gel is made up of some other substances known as acrylamide. Okay, so that is for this as a separate information. You can remember we call it as page. Okay, this one we call it as age, agarose gel electrophoresis. This is called as polyacrylamide gel electrophoresis. This is mainly used for protein separation. Okay. Then another question that comes in the exam is, let us say after doing this, let us take any one example. Suppose you have taken your sample here, right? Sample means what was the sample? You have taken the DNA earlier and cut it down, and after cutting the DNA, multiple fragments were formed. Now your idea is to identify a particular fragment which is of your interest. So let us say when you have put this sample, you came to know that this is the DNA band which contain your gene of interest. Now the point is, how are you going to separate, because you need these fragments now for further experiment. So what is done is very simple. With the help of some kind of a cutter, you can cut off the gel portion containing that particular band. Let us say this one. So you cut off this particular gel portion containing the band, and then you have a technique wherein you will heat it up so the gel will dissolve, and then you have to purify the DNA from it again. So that technique is called as gel elution technique. DNA bands can be cut out from the gel and DNA can be purified from it. That is called as a gel elution technique, by which you can obtain your specific gene of interest from the gel after its separation. Now there is one thing just a minute, let me take out a picture from somewhere. In the meantime, if anyone has a question, please tell me. Did you understand about agarose gel electrophoresis? Anyone has any question? Please tell me.
Sir,
Yes, tell me.
so can you explain how the minute pores work?
Pores are formed because of cross-linking molecules. In the gel, those agarose molecules will form cross-linking. Okay, so that is why in between the molecules there will be small pores that will be left out.
So do the DNA molecules get like impregnated into these pores?
Sorry, DNA molecules?
Do the DNA molecules get accumulated into these pores?
Not accumulated. The thing is, the DNA molecule will continue to move through those pores. So when they move through those pores, based upon the size of the molecule, they will get stuck in certain pores. Okay, either they will move. If they are very small, they will continue to move forward. If they are larger molecules, they may not move further forward, and they will get stuck there only. Now see here, Let us say, for example, A and B, you leave it. Let us say C, in this well, you have pushed your DNA mixture. The DNA which was cut down to get 3 fragment size. Okay? To get 3 fragment size, out of which 1 fragment is your gene of interest. So these 3 fragment size, therefore, you will see that they have settled down at 3 positions, 1, 2, and 3. So one thing you have understood, that the 3 fragments formed by cutting the DNA molecule, those fragments are separated from each other. Now the big question is, how will you identify which fragment is the fragment of your interest, or which of these band contain your gene of interest? Because you can only see their separation. We know that they got separated based upon their molecular size or fragment size. Now the point is, which of the band contain your gene of interest? Because you cannot see any nucleotide sequence here. It is only that the DNA is, fragments are separated and they are settled at different position in the gel. So for identification of the band containing your gene of interest, we normally use something called as a marker. Okay. Now what is a marker? Please understand. We make the gel in such a way that in one of the well we will put the marker, and in rest of the well we can put the sample. Now let us say this is the marker. Marker contains, it is a mixture of multiple DNA fragments, okay, of, very important, known molecular size. Okay, known molecular size means through various studies, research, it was found out that here this marker is made by mixing the fragments whose size is already known, and therefore when it is separated, it is known that the one which will move maximum distance will be 100 base pair molecule. The one which will be second from the largest distance will be 200 base pair, and so on. The one which will move the minimum distance will be 1200 base pair fragment size. Now this marker, also sometimes called as ladder, okay, this is also called as sometimes ladder DNA. So this ladder DNA or marker DNA helps us in comparing the fragments of our molecule which are of unknown size. When you do these experiments, these markers are ready-made, found in the market to be purchased. Okay, when you purchase it, there will be already a diagram or an information mentioned in the package, that when you will run it in the gel, there will be these many 10 or 12 fragments will be found, and their relative, based upon their relative distance, you can compare that the first one will be 100 base pair fragment, second one will be 200 base pair fragment, third one will be 300 base pair fragment, and so on. Now what you have to do, you have to know what is the size of this fragment in this band. So you compare, and we know that at the same position, the same distance moved by the known molecules contain 600 base pair size fragment. Compare the second one, we understand that this fragment of our DNA will have 300 base pair sequence. Compare the third one, we understand that the last fragments will be 100 base pair fragments. Let us say that we know that our gene of interest is a 300 base pair fragment. That is something we already know because we know about our gene of interest, right? That is, we have done the research and found out. So it means that this is the band which contain my gene of interest. So I will do the elution of this band. I will cut off this part of the gel and separate the DNA from it. So the principle is irrespective of the nucleotide sequence, whatever is the nucleotide sequence, based upon the fragment size, the separation is happening. So we have taken some type of other DNA molecule whose fragment size is already known to compare the DNA molecule of our interest whose fragment size is known but we cannot identify where it is located. Okay? So this is the technique which is not there in your book directly actually, but this is the technique by which you can identify the specific band containing your gene of interest and isolate it. Therefore, from here, when you do the gel elution and take out the DNA, this DNA is your gene of interest which you want to play with in the remaining experiments. Is this understood? Anyone has any question? Please tell me. That is all about gel electrophoresis.
Sir,
Yes.
sir, like out of so many base pair, how the researchers like they have
What?
how they like they find that this base pair is like they needed for like gene of
So that is see, that is why we say that these are applications. Okay, this is the application of what we have already learned. This is not the preliminary research. So these base pair, how many base pairs are present in a particular fragment, that is done by research, that is done by studying through other techniques. There are techniques by which you can identify the specific not only number of base pairs, but you can also identify which are the bases which are present. There are sequencing techniques. You will learn about some of the sequencing techniques here. Okay, the sequencing techniques are there. Sequencing techniques means to identify the sequence of nucleotides. Understood? There are techniques by which a particular fragment, what is its sequence? Maybe someone has, what they did is they have taken some DNA, broken down that DNA into multiple fragments, and they have run it into the gel, and they got all these different fragments here. Maybe they have taken the fragments from each of the bands and they have done sequencing. Each of the bands they have taken and they have done the sequencing. When they did the sequencing, they found out that how many base pairs are present in these bands and which are those base pairs. Right? So that is a technique. Now we could have done the same for our DNA mixture also, but the thing is that will become very cumbersome. That will become more time consuming, more costly. So based upon something which is already done and known, we are carrying out an easier technique. So that's why we call these things as applications as such. Anything else, anyone? Okay. So the next topic is something very important. Vectors. Okay. Like I gave you some brief in the previous class. We were talking about an example of making insulin protein. How it is done? We obtain human DNA. We cut the insulin gene from the human DNA and we put that gene into a bacteria. Then the bacteria will start producing the insulin protein for us in large quantity. Now the point is, when you put the human gene inside bacteria, bacteria will not accept it. Okay? To make the bacteria accept that particular gene, you need a vector. This vector will be a kind of carrier of the foreign gene. So this will carry the foreign gene into the host. Okay, so the vector is very, very important. What type of vector are you going to use? That usually depends upon the host. Okay, when the most commonly used host is bacteria, right? And for a bacterial host, the most commonly used vector is a plasmid vector, right?